the first speaker is Latifa Jackson, and the second one is Rosemary, Bastian. At the end of these presentations, there is a short couple of slides about current research projects going on at our school, which are looking for students to join, both two labs, which are looking for master's and PhD students to join various projects. So without further ado, Latifa is actually, believe it or not, second-year PhD student who will be graduating during the summer term. One of the quickest PhDs. She is working with Dr. Turner in bioinformatics and genetics on a population level, and she's going to talk to me about her research.
Thank you, guys. So I'm going to talk with you really briefly about something that gets me up in the morning and gets me into the lab and gets excited. And you know, from all of you guys' dissertation work, no doubt, that sometimes you can feel like you're the only one who's super excited about your project. I'm super excited about this project, and I think it has some implications that are far-reaching. So my talk title is Addiction, Mental Health, and Infectious Disease. Let's see if I can effectively use this. What is addiction? Let's start there, because I like to start with everybody being on the same page. So addiction is loosely defined as a chronic relapsing substance disorder. It's characterized by loss of control over substance agents. So as you see, the definition is pretty amorphous, right? It's a behavior-based phenomenon with a diverse array of psychological, biological, and genetic contributors. And then, of course, it has environmental and cultural factors. And the genetic components of substance addiction, in particular, because there are these huge environmental, psychological components, are not fully understood, I should say. Although we have some interesting things to say about that. So, while studying it, obviously there's plenty of people in this country are looking for substances, and they cost $500 billion in health and judicial services to the U.S. government. And then in addition, I don't know if you guys recently saw, I think it was this morning, the BBC came out with a report that said that there's so much cocaine being used in the UK that actually when they do samples of water, that there's actually cocaine in the water. That could be the new vitamin water. So these substances have different kinds of effects, and they can be characterized generally by their activity family, which I'm going to talk about. So let me stop there and tell you kind of where this talk is going, and I like to tell you where it's going by telling you what questions I'm going to ask, and then we're going to address those questions. So the first one was, there's been an increasing number of association studies, and if you don't know, association studies are basically where they put a particular gene, and then they say, I think this gene has something to do with a particular phenotype, a particular disease trait, and then they survey that in a population of people who have the trait and who don't have the trait, and they try to understand whether there's a strong correlation between having the trait and having some particular variant at a gene site. So I'm trying to understand, of all of those genes that have been previously identified in the literature through molecular work, through functional work, through association studies, what role do they have, the gene locality have in addiction disorder? And then second, how does the variation at these potential sites vary in human populations? And then the next question is, what is the intersection of addiction and mental health conditions? Because remember, at the very beginning, I told you that there's a huge psychological component to addiction. And so we really want to try to understand that. And then last, we really want to try to get at one of these classical kinds of questions in population genetics, which is that this concept that infectious disease drives a lot of the variation that we see in human populations. And so we're going to try to get at that. So without further ado, I'm going to start showing you pictures of data, which are not as exciting to you as they are to me, but what we did was we took all of the genes that have been identified as being associated with addiction. So these are three types of addiction. There GABA, which is alcohol and GHB. which is the date rape drug, just in case you're not up on your drugs. Then dopamine, which was cocaine and crystal methamphetamine, and then opiates, which was heroin and morphine. And I really tried to focus mostly on illicit drugs, although you could potentially add more drugs, and that's something that I've thought about as an extension to my dissertation work. What we found, interestingly, was that if you take all of these genes, now we're talking about 600 genes that have been previously characterized as being involved in these disorders, of which only 17 are shared amongst these genes, amongst these three classes of genes, that they have so they have 7 hotspots in the genome for which we see an overabundance of genes. And so you're like, well, how do you know? So what we did was we ran simultaneous simulations to try to understand how many genes, because obviously genes have to sit somewhere in the genome, right? So in order to try to understand whether what we're seeing is something that's overabundance or whether it's something that is just normal, we ran simulations, and the p-values are actually 0.005, so significant. 4 of these hotspots are associated with all 3 drug classes. That is to say that genes contributing separately to all 4 of these drug classes are found at these hotspots. And then 3 of them are specifically associated with GABA addiction, and in particular, really it's alcohol addiction that's driving the GABA addiction profile, because the large majority, 90% of those genes, are actually coming from alcohol addiction. So the next thing that we wanted to do was to try to survey these in human populations, because we know from the biomedical literature that a lot of studies have been done on American populations, African American populations, European American populations. And so both the predominant number on European American populations. And so we wanted to try to get a sense of what's happening. And here, once again, we can use population genetic approaches and evolutionary biology approaches to try to understand what's happening at these particular regions that I've now outlined to you. So in these 7 regions, we then went to the HapMap data set, which is about 1,000 individuals and 11 different populations. Here's their geographical spread. And this data set has 10 million SNPs sampled across these populations. So let me stop here before you're like, oh, there's only one signature, which is this delicious one here. Let me tell you something. So first, before you can identify what you think is happening in the population, we need a baseline, right? The same way that you use a standard in a molecular experiment, we need a molecular standard, if you will, for a genetic sample. And in order to do that, I looked at 20 autosomal regions. Those are non-sex chromosome regions all across the genome that have been shown to be neutrally evolving, and tried to characterize what the patterns look like in those regions, right? So in neutral regions, the only signature that we should see is nothing selection-based. We should only see the effect of population history. So that's what this neutral comparison here is. And as you notice, it's a pretty dark square. And darkness in this example equals non-significance, right? And so then we were able to make comparisons at these particular locus regions that we were interested in, and the regions that I'm in particular calling addiction hotspots. So you see here in particular this characteristic cross pattern, which is just basically saying that East Asian populations, in particular here, the Chinese that were sampled in Beijing, Chinese that were sampled in Denver, and Japanese populations show significant allelic differences from the block of African populations here, the block of European, South Asian, and Mexican populations here. So when we discovered this, I mean, and then there's a secondary pattern that's much weaker, which is that at this chromosome 4 site and at this other chromosome 6 site, that an African population, a West African population collected in Nigeria, has significant allele frequency differences from all other populations, basically. did this, we actually were able to identify the polymorphisms that are driving that pattern that we saw. And once we collected that list of polymorphisms, and then we conducted what we used the GWAS 3D platform in order to understand information about the long range and local linkages. So we know from when we initially started that we started out with this huge gene list. Some proportion of those genes ended up being in hotspots, and then from there we identified particular polymorphisms that were important at those hotspots, right? So now we've gone from genes to regions to particular polymorphisms. But now we really want to make sure that we make a little bit of a connection back and say, well, what does the genome-wide association literature say, right? And so by entering in these significant polymorphisms, we found something really kind of interesting, and that was that we actually returned some of these long-range, so all of these are showing long-range interactions, like the ones that go all the way across, and then the local ones are like here. And what we found was that we actually returned some of the initial genes that were on the list. So say, for example, this MAOA gene was not actually one that clustered into a hotspot, but it has a long-range interaction with the particular polymorphisms that are significant in African populations at that hotspot, right? And so we think that this is really interesting, both in terms of using it as an approach to identify important candidate polymorphisms, but then in addition to really understand the long-range interactions that underlie these genome-wide association studies. And so we think that this is a result that bears further investigations. So just quickly, I want to remind you what I just went over, which was that we identified these 7 hotspots. 3 of them were alcohol specific, and then 4 of them included all of the other addiction classes. That these, I forgot to tell you, but they're functionally similar. So when you look at the genes that have been identified in the hotspot, they also share the exact same functional annotation with the neighbor genes that have not been characterized as being involved in addiction. So what we're seeing here is not just the genes themselves, but they're actually, the whole region is functionally uniform. In East Asian populations, we see this cross pattern, which shows that there's different allele frequencies in populations at this histone-rich region. And then that at these other 2 regions, we saw a pattern where African populations had significant population differences from all other populations. But this region was interspersed with immune genes, and so that kind of motivated the work that you're going to see in the 3rd question, because this was definitely a kind of a rolling dissertation in that sense, that you kind of find results and you think, I have to follow this up, this is too intriguing. And especially for someone like me who has a population biology background and not so much a biomedical engineering background. So on to the next question, which was, what was the genomic intersection of addiction and mental health? So, substance addiction and psychological disorders, as I've said, have been previously characterized and commonly characterized as comorbidity. And there's been shown in the literature a reciprocal relationship between alcohol dependence and depression or schizophrenia and cocaine use. We see a whole series of these articles that basically go back and forth and kind of recapitulate this question about the chicken and the egg, which comes first, right? And honestly, alcohol is often characterized as a psychological disorder, and it was this basis that led us to really ask, what is the intersection of these 2 disorders, right? And so each of these disorders have been well characterized individually, but not necessarily well characterized together. And so in the literature, while people have the chicken and the egg question, they don't get down to what that actually cycles out to in terms of biomedical relevant markers. So in particular, I look at three disorders, and those are schizophrenia, bipolar disorder, and depression. Each Each of these has a relatively significant prevalence in human population. So 1% of the global population is 1% of 7 billion. 2.6% of American populations for bipolar, and 17%, that's 1 in 6 Americans suffer from depression. So I think that if you probably think back to, especially as grad students, if you think around you, you probably know somebody who suffered from depression, not saying bipolar or schizophrenia. But one of the things that these all have in common is that they have strong genetic components, which is really important because, as you see, my work is computational and it's not molecular, so I'm not in the lab trying to head out the answer. But collectively, these 3 disorders themselves represent one of the top 10 diseases, more important than heart disease in terms of life years lost. So when we took the combined set of all of the genes that have been curated through NCBI as being involved with these 3 mental health conditions and the set of genes that we had previously identified as being involved in addiction, we identified 8 genomic regions across the genome. And then we found 3 regions that are specific to bipolar that don't match to any other disorders. But all these other ones, the ones that say all, are all regions where genes from addiction, from bipolar, from addiction, from schizophrenia, and from depression are all located. Interestingly, when we did this analysis, we actually found 0 hotspots that are related specifically to addiction. So when you take the whole addiction gene set and look at them across the genome, nothing. And that makes us question what addiction truly is in terms of its cohesiveness as a disorder, although that's a question for another day and another dissertation. So the next thing we did was not to look at human populations, because my goal in this was not to show that I could once again identify polymorphisms that underlie this trait, although we know that we can, but to try to understand what drug targets were sitting in these particular regions, because another way to validate these regions as functionally important is to try to understand what drug targets are located there and to annotate those. And when we did that, we found 16 drug targets at 5 of the 8 hotspots. And these drug targets basically fell into 4 main classes. And I'm just going to read them to you because I realize it has a little bit small. Those ones were addiction, so binding sites for ecstasy, for cocaine, binding sites for mental health conditions, for pharmaceutically approved drugs for Parkinson's, for PTSD, and for anxiety, right? For infectious disease, once again, we're seeing this infectious disease kind of rise up, and so for influenza and for hepatitis B. And then finally, there was a whole bunch of cancer drugs, and I was kind of like, what does this mean? Or are there just cancer drugs everywhere? So I went in and I tried to look at the medical reports of these cancer drugs, and interestingly, the counterindications for these cancer drugs, so you know how like, if you listen to one of those like, I don't know, priapism, things that's like, you might die of these other things. So in this case, the counterindications are anxiety, addiction to these substances, and those are not consistent with the other cancer drugs that are in these class. So there could potentially be some effect that this region is having on these cancer drug binding sites. So once again, this recapitulates the idea that there's a linkage between addiction, mental health, and immunity. And I would suggest that that's probably a paradigm that we have not we have not coherently thought about, because all of these topics and subjects have been really studied in silos. So just really quickly, I found these 8 hotspots that sit at the intersection of addiction and these 3 mental health conditions. And then there will be 16 binding sites that represent 5 hotspots that are responsible for the treatment of addiction, mental health, and infectious disease and cancer. And finally, I went to infectious disease, and here, so let me ask you guys, how many of you guys have ever studied population genetics? I feel like I've been throwing that around a lot. Anyone feel it? Okay, cool. So you don't need to, but I do know that Don McEachern teaches a class in evolutionary applications for biomedical engineers. So if you ever are interested, you might want to take that. I feel like I should plug his class at this moment. So one of the fundamental things that came out of this idea of mapping, sort of Darwin's idea that there's lineages and that they persist through time, and then Mendel's work that there's genetics, and genetics are the linkage between the lineages. When those things came together in the modern synthesis, one of the first things that they identified was that, oh, okay, infectious diseases must be one of the strongest forces in human history in terms of shaping genetic variation in populations. That is to say, if you don't survive this year's malaria outbreak, you're not going anywhere. Your lineage is not going anywhere. And there's one thing that's consistent with everyone here in this room. You come from a lineage of survivors, right? Right? Okay. So this was identified early by work with Howden in malaria, and then further studied by Allison. But one of the things that's really interesting about this effect is that you can have some particular allele, some genetic polymorphism that is confers some sort of higher fitness. So this is what's underlying those association studies. But then what happens to all of the variation that's around that, right? And because of this idea of linkage, or linkage disequilibrium, we know that we can actually see an increase in alleles that are not particularly, that don't confer any fitness benefit, or potentially could be slightly deleterious in terms of their effect on fitness, right? But they're rising in frequency over time because this red allele X here, as you can see, after time has gone on, it confers a fitness effect. So, for example, having a really strong immune system may lead to an overabundance of people with blue eyes. Do they have anything in common? Nope. But if the two traits are close together, and eye color is actually an erroneous trait because it actually is multi-locus, but let's say Tay-Sachs, right? Then you can see an increase in those frequencies even though the trait has negative effects. And so we wanted to really consider what role infectious disease has in driving the risk alleles and the resistance alleles that are identified both in cocaine, heroin, all of these basic addiction classes. Because obviously it can't be very advantageous to have high susceptibility to cocaine. That just can't possibly be, right? So the question is, why do we see them in the allele frequencies that we have, that we do see them, right? And association studies have shown that, particularly in African American populations, we see these like, high frequencies in African-derived populations, and our initial studies kind of bore that out a little bit. So the Yoruba, who are in West Africa and Nigeria, are one of the source populations for African Americans here in the U.S. So while the work that has characterized the susceptibility has been done in African Americans, we can use this finding in the Yorubans as a good indicator that this is African-based. And then, so what I did here was we tried to identify, once again, those hotspot regions. I feel like this is kind of like the core of the method, because what we're really doing here is reducing the number of sites that we have to survey in the genome. So nobody wants to look at, 3 billion polymorphic sites. Instead, we can narrow that down to, 200,000 or 300,000 polymorphic sites. And we can use this approach, which has been shown to work in the first and second question to do that. And then in addition, we wanted to expand out the population set that we looked at. So before I looked at 11 populations, now I'm adding 51 populations. Here they are. So what we have here are populations that are living in temperate climates, populations that are living in tropical climates, tropical populations that are living in mountainous regions and non-mountainous regions, and different ethnic populations, right? And we can use this information to ask two questions. And those questions really are first, what is the effect of tropicalness? And we're using tropicalness as a proxy for infectious disease because there's a strong correlation between the actual environment that populations live in, the long-term evolutionary environment that populations live in, and their infectious disease burden. And so we can do that with American populations. So this is like a population that's in right outside of Tucson. Obviously, they're not sitting in a tropical environment, they're sitting in another environment. We can do that here, where we have an Algerian and Assam population that's sitting on the outskirts of the Kalahari Desert. So obviously they're not in tropical climates, and then here we have populations that range from tropical Southeast Asia up to the tundra of Russia. And so we can look at these different populations and try to get a sense of what role temperateness or environment plays as a proxy for infectious disease burden on human populations. And remember that we're characterizing now only the intersection of genes that are sitting between immunity and addiction, right? So we're not just looking at immune genes. And when we do that, you get this crazy graph that means probably nothing to you, but what you can hopefully see is that things track pretty well. So I found 3 hotspot regions. In these 3 hotspot regions, there were 3, these 2 regions here, where there were polymorphisms that were significantly different. And this is just a quick way to visualize that. But let's go into what those actually mean. So when we sorted these populations, we sorted them into high tropical, low tropical, and low tropical meaning temperate, i.e. no tropical. And then we identified these 5 polymorphisms. And when we identified these 5 polymorphisms, there's a whole lot of method stuff here. You guys don't want to hear that. That's like for another day. When we identified these 5 polymorphisms, we found three sat in open reading frame region, which open reading frame regions are notoriously poorly characterized, but this open reading frame region is the only identification of it is that it's involved in the liver interactome. It appears to have some potentially xenobiotic clearing function. Xenobiotics are like drugs, like cocaine. And then one sat in this amber 1 region, and this one has previously been characterized in 2002 as being strongly associated with schizophrenia. And it alters, and then in a subsequent study, they actually took this polymorphism and did a behavioral analysis and identified that it's involved in impulsivity-related behavioral and neurological traits. And then the final one, interestingly, is involved in repair of UV damage to DNA. But, so that sounds kind of amorphous, and when we look into the association literature, we actually find that it's a critical gene that forms a complex that destabilizes the hepatitis B viral protein X, right? So without this gene being overexpressed, you actually don't have proliferation of hepatitis B, right? So it has strong immunological implications. So, but all 3 of these ones are sitting relatively close to each other, and we think that they bear further study. Okay, so finally, I just want to remind you that we identified these 3 hotspots, which we knew the technique worked, but we wanted to polarize the populations by environment and really understand how environment and in particular infectious disease play a role in identifying candidate SNPs. And what we found was basically, once again, this paradigm where mental health conditions, addiction conditions, and immune conditions appear to have much more crossover than we understood. So with that, I just wanted to acknowledge my lab, my advisor, Dr. Natalia, my research group, Max and Javon, who are here, the bioinformatics lab upstairs, who are amazing, and my funding. I have a Dan fellowship and my team members. That's it.
Rosemary Bastian will be your next speaker. Rosemary works in Dr. Chaikin's lab, which is the Drexel College of Medicine. She's advised by Dr. Chaikin and Dr. Ian Chef, who is her biomedical advisor. And her work is in HIV.